glucokinase. You should have known it was a kinase, so you should have known A and B were wrong, right? It said in the question it's a phosphorylation of glucose, so you should have known it was a kinase. Phosphofructokinase doesn't make a lot of sense, right? Not for glucose. Probably does something phosphofructose. You laugh, but I promise you'll see questions that are beyond your. Not all of them. You will see some questions. Okay. We kind of flew through reaction 4. So we ended on Friday. And we have a long way to go because we are going to finish today, all the way through today. That is our goal. We can cheat on a few of these and say it looks like that, it looks like that. We kind of did that on number 4, right? I told you there are 2 classes of enzymes, class 1 and class 2. Class 1 and higher organisms, class 2 and lower organisms. They differ. One of these forms a covalent enzyme substrate intermediate. The other one doesn't. The one that doesn't, how does it stabilize this intermediate? Medical factors, right? One of the main use of medical factors, biological organisms, is for stabilization of vital function. So this reaction leads us to 2 products, right? If we're going to cleave our sugar, it's going to lead us to 2 different products, right? Yeah, this is kind of exciting. It's going to leave us with 2 different products, DHAP and G3P. We want G3P. So how do we convert our next reaction? Reaction number 5 converts DHAP 3. Reaction number 5 converts DHAP to G3P. And this is the one with the G. This is an isomerization reaction. As much as I would love to draw this, and I'm not being sarcastic, I really would like to draw it. I told you I like to see double bonds in it. I can see double bonds in it. We're not going to draw this. Why are we not going to draw this? Because it looks exactly like the isomerization reaction we talked about on Friday. This reaction is catalyzed by triose phosphate. What type of enzyme is it? Triose phosphate isomerase. And you may be, in a second we're going to talk about another isomerization reaction. It's not labeled an isomerase. Why? Because isomerization is a very, very broad, definition. There are different types of isomerization. So you'll see in a second that we don't call this our other enzyme an isomerase. It is an isomerase. It's just all dehydrate or all. Remember we have a oh my goodness, dehydrogenase enzyme that we talked about earlier. It is an oxidoreductase. You don't call it oxidoreductase. Dehydrogenase. It's one enzyme. It's a subclass, I guess you could say. Subclass. That took way too long to come out of my notes. You should be able to draw this mechanism, especially after you've drawn the mechanism in step 2. So if you have trouble drawing this mechanism, go back and look at step 2. The only difference is the direction. The direction is different because you're going from ketose to maltose, right? Intermediate, diol, intermediate. And that completes the priming phase, the preparatory phase. That completes the preparatory phase. From here on out, we're going to start generating things that lead to the production of energy. So first step, why DHAP? Why do we need to convert DHAP? Because the next step oxidizes G3P, not DHAP. So now that we are dealing with remember we have 2 moles of this product for 1 mole of glucose, right? Our next step is oxidation G3P. The oxidation of G3P is a coupled reaction. Oxidation reaction, so we're gaining or losing electrons from G3P. We're losing electrons from G3P. Those electrons go somewhere. Talk about some coenzymes that are involved, specifically evolved to transport electrons. Oxidation G3P is coupled to product 1 through VPG should look familiar. Where have you seen VP? Where you listening to people going, right? Facilitated the release of oxygen or hemoglobin. Was that 1,3 VPG? 2,3 VPG. It is a regulatory molecule that allosterically binds the hemoglobin, favors peace. Doesn't bind oxygen. So we have oxidation of G3P to 1,3 VPG, and this is coupled to the reduction of NAD plus to NADH. And the enzyme does this. G3P. What class of enzyme is this? What class of enzyme? Oxidoreductase. Oxidoreductase. Those are enzymes involved in redox reactions. Enzymes involved in redox reactions. It's an oxidoreductase. Oxidoreductase is a broad name for all different enzymes, right? Dehydrogenase is a specific type of enzyme involved with transfer of a hydrogen, right? Get it? Get it? Sorry. So G3P. First step is nucleophilic attack from my thiol. Where is my best electrophile? Carbonyl carbon. Five bonds on the carbon. Leads us to a enzyme, a covalent enzyme substrate intermediate. Let's draw our NADH first so I don't run out of room. This is NAD plus. Enzyme associated complex. What's up?
Are you missing that O after the CH2?
I am. Did I do it over here as well? No. Thank you. I was gonna say to make it look nice, but this isn't making it look nice. Thank you. So this leaves us with immediate. So we have reduction of NAD to NADH, leaves us with an intermediate, and this is a unique part of this reaction, a unique part of the entire process. This is the only point in which inorganic phosphate attack, nucleophilic attack on a carbonyl carbon, an inorganic phosphate leads to final products. So this differs from phosphoryl transfer. Why? Because both of the phosphoryl transfers we've seen have required what? In the first 5 steps, we had 2 phosphoryl transfers. Both of them required something. What?
What happened to the PO3 to minus in the bottom part?
It's right here. Right there. It was hiding. Thank you. It would be BPG then, right? Question, who answered? Somebody did. It doesn't require ATP, right? So here we have phosphoryl transfer, not really phosphoryl transfer. Phosphate transfer, right? A phosphate transfer. It does not require ATP, and it also yields a net gain of 2 NADH. So why 2 NADH? 1 NADH. However, 1 glucose leads to one G3P, one DHAP, one DHAP goes to G3P, so it actually leads to two G3Ps, two G3Ps. So one one glucose will lead us to two NADHs by this step. Two, yeah, NAD. Sorry. Two last points. One, reduction of NADH, NAD plus to NADH, also is known as a term called hydride transfer. You see a hydride transfer reaction. Movement of electrons. How many electrons? The phrase hydride transfer should give this away. Hydride transfer. What is a hydride? If you don't know, let's count our oxidation states. What's the oxidation state of our carbon that's changing oxidation states here? Oxidation state of this carbon is -1, 0, +1 is a net of +1. Oxidation state of the same carbon for my end product. That is +1, +2, +3. Instead of this part, +3. How many electrons do we lose? We lost a proton as well. So 2 electrons and a proton. That's a dehydrogenase, isn't it? That's a dehydrogenase. 2 electrons and a proton. Dehydrogenase. So this is a 2 electron oxidation. Oxidation of a reduction of NAD plus the NADH is a 2 electron hydride transfer. 2 electron hydrogen transfer. Next step is transfer phosphorus group. Transfer of phosphoryl group away from, so that's kind of important. All of the transfers we've been doing are towards our substrate, right? This is a transfer away from our substrate. Phosphoryl transfer away from our substrate. Away from 1,3 BPG. Away from 1,3 BPG. What kind of enzyme? How would we name it? An enzyme involved in phosphoryl transfer. Kinase, right? It's a kinase. Phosphoglycerate kinase. Transfers our phosphoryl group to an ADP, which leaves us with ATP, right? So this gives us a net gain of 2 ATP, right? And ATP. Net gain of 2 ATP. Why? Because we have 2 1,3 BPGs broken down glucose. So this is a net gain of 2 ATP. This looks a little weird. So 1,3 BPG. At this point has a choice. It doesn't have a choice. It involves 2 pathways. One pathway, the one we just talked about, phosphoglycerate kinase, leads to 3 PG, 3 phosphoglycerate plus ATP. The other path we're going to follow is BPG mutase. What do you think of mutase? It mutates it. What is that? Mutase, a mutation reaction in terms of organic chemistry is just a movement, right? It is a class of isomerization reaction. So a mutase is an isomerase. Sure, we don't label it the enzyme name an isomerase. Why? Because it is a mutase reaction, which is a specific movement of a functional group inside of a molecule. So if we're going to move a functional group, so if we're going to move one of our phosphates, specifically, we're going to move phosphate 1 in the first position to the second position. At this point, we would be left with 2,3-BPG. Yeah. Kinase. PG kinase. So 1,3-BPG, thank you. 1,3-BPG can follow two pathways, right? Phosphoglycerate kinase is involved with the formation, the direct formation of 3-phosphoglycerate and an ATP. It follows pathway with BPG, this phosphoglycerate mutase. It is a mutase reaction in which the phosphate group moves from carbon 1 to carbon 2. Moves this with 2,3-BPG. At this point, 2,3-BPG phosphatase and complete the reaction to 3-phosphoglycerate plus inorganic phosphate. So regardless of the pathway, we are led to the same product, 3-phosphoglycerate. It can continue on. 3-phosphoglycerate is going to be the next step of our reaction. However, there's a difference. Main difference being one produces ATP, the other doesn't produce ATP, right? But we've already talked about, does this serve a function? Does this intermediate step, it's not an intermediate of a single reaction, but it is an intermediate step of a pathway. Does this intermediate step, does it serve a function? We've already talked about one function. It does serve a function, right? What does it do? It regulates hemoglobin, right? It regulates the release of oxygen of hemoglobin into muscle tissues, right? So it does serve a function. We've already talked about one function it serves. It also serves another type of function that we'll talk about in the next step. However, we're going to talk about two ways to produce energy. So our first way to produce ATP, energy in the form of ATP, is called, well, they're both called phosphorylation. First is called substrate level. Substrate level phosphorylation. Substrate level phosphorylation is an example of what we just saw. We take our substrate, we transfer a phosphoryl group from our substrate. So the transfer of a phosphoryl group to an ATP is driven by the substrate. It's called substrate level phosphorylation. It produces a small amount of energy, one, right? However, this is not your textbook, and we'll get into it eventually. I think in both textbooks it's going to talk about this. It's going to kind of give you a little brief primer. We're going to kind of gloss it over until we get to our last chapter, which would be oxidative phosphorylation. Or respiratory phosphorylation. Some texts may call it respiratory level phosphorylation. Oxidative phosphorylation, respiratory phosphorylation in the presence of oxygen. So a pathway in the presence of oxygen, we'll use NADH, flow of electrons to generate phosphorus. We'll talk about it later. These are not the same thing. One of these produces energy much more efficiently than the other. They both take place in different parts of the cell. One of them utilizes substrate, the other one utilizes flow or gradient of electrons. We'll talk about that. Now what song is that? Is that Coldplay? Yeah. That's what I thought it was. Is it Coldplay?
I think so.
Okay. Yes? That's what I thought. But then anyway, I like it. Next step. Great, you got 20 minutes to finish these last 3 steps. Perfect. Conversion of 3-phosphoglycerate to 2 phosphoglycerate. Conversion of 3 phosphoglycerate to 2 phosphoglycerate. This is catalyzed. By PG mutase or phosphoglycerate mutase. Hopefully this is starting to make sense of naming conventions of these enzymes. We just talked about conversion of 1,3-BPG to 2,3-BPG uses BPG mutase. I told you, knowing that information, that we're trying to convert 3-PG to 2-PG, you could assume we're going to use another mutase, right? Phosphoglycerate mutase. Hopefully the naming convention is starting to make a little more sense. Phosphoglycerate mutase is kind of unique because it requires an active site on our enzyme that has been phosphorylated. Phosphorylated enzyme. Phosphorylated enzyme. Requires a basic amino acid like histidine. Protonated histidine, but it's not been protonated with the proton. It's been protonated with the phosphate group. So it's a phosphorylated enzyme. Another amino acid, basic amino acid. Reaction started by general base catalysis of our histidine, resulting oxyanion is able to grab our phosphoryl group, which leaves us with an intermediate that is going to look similar to something, at least something we should recognize when we're not seeing it. How would you name that intermediate? How would you name this? What is our starting material? 3-phosphoglycerate. How would you name this? 2,3-bisphosphoglycerate. So this intermediate is actually, so you don't hear this very often. Very often. You don't hear this very often. So the intermediate of this reaction is, it's still in an active site, right? And if you're wondering what this green stuff is, this is our enzyme, right? So this is our active site. These are active site residues of our enzyme. I didn't say that, so we can do that. Should have said. So this intermediate, it's weird because you don't hear this phrase a lot. You don't hear the phrase as far as enzymatic reactions. You see this in organics now. As far as enzymatic reactions, you don't hear this phrase a lot. You're going to see it here. Phrase stable intermediate. Stable intermediate. You don't hear it a lot, right? You always hear unstable intermediate or destable intermediate. Something that's very reactive. That's the point, right? You get it to a point where it's reactive and then it can react and it can go. However, 2,3-BP 2,3-BPG, is a stable intermediate. You know it's a stable intermediate because we've talked about it before. It serves a function, right? So at this point, 2,3-BPG, while it will, for the most part, continue along the pathway, 2,3 can readily associate or dissociate. I'm sure this is very controlled, and the whole point of an enzyme is to not let an intermediate dissociate, right? That's the whole point of an enzyme, is to facilitate catalysis all the way through, not halfway through, and then release intermediate. But it can. It is not very effective. I think your textbook, the Biochem textbook at least, says it's usually out of 1,000 turnovers, BPG will release. So it's not often, but it can release. So, why do we care? Let's keep going. That'll make sense now. So, got something. We got chemistry, right? The whole point is to get to 2 PG, right? 2 PG. Base. Grab our phosphorus group. So if you're not CI, grab proton. We are left with phosphorus group, 2 position. We're left with a phosphorus group with a 2 position instead of 3 position. And you'll notice, what does this do to our resulting enzyme? Regenerates, right? So this is going to be plus regenerated. Regenerated enzyme, right? So at this point, our enzyme requires something unique, right? A phosphorylated amino acid at the active site. Turnover from this enzyme is actually going to re-phosphorylate that amino acid at our active site, right? So let me ask you, what primes this to start? If this requires a phosphorylated enzyme, you can imagine probably, I have never worked with this enzyme, so I don't know much about the replication and the translation of this enzyme, but I bet it's not translated with this modification. I bet it isn't. It could be, and I may be wrong, but I don't think so. I bet it's not translated with that modification. How could that modification occur? What? How could this modification occur? Assume we never had this step right here. Assume this never happened. So we're not going to go from 3PG to 2,3-BPG. How could we initially prime that enzyme? So let's assume we're at this state. At this state, there is a resting state of the enzyme. If this enzyme is not phosphorylated, is this a reasonable resting state at neutral pH? Why sure? Because the pKa is pretty close, right? The physiological pH, the pKa is going to be relatively close, that you can assume that there's going to be a certain portion of histidine that is deprotonated and a certain portion of histidine that is protonated. If at this point, if this is our, it's not our resting state of our active enzyme, but it is a resting state of the enzyme. At this point, if BPG can associate and dissociate freely, imagine if we have 3-BPG produced from the previous step. Sorry, 2,3-BPG produced from the previous step. We said that it doesn't serve a function as far as energy production, but it can serve additional functions, right? I already talked about 1, regulation of hemoglobin, but 2, it can serve as an activation of the phosphorylation of our enzyme for our next step. 2,3-BPG could associate, and it can start this step, not at step 1, but actually at step 2. Does that make sense? It can actually start this, phosphorylation of enzyme from enzyme that's been deactivated because it needed a specific cofactor, right? It needed that modification, that phosphorylation, this histidine. Let's say we lose that phosphorylation of this histidine, or it's not generated during translocation or replication of protein. If it's not generated during replication of protein, this reaction can be primed from the intermediate step by 2,3-BPG. Does that make sense? So if this somehow messes up, it somehow gets turned off, if you somehow lose this phosphoryl transfer to something else, if this phosphoryl transfer does not get back here, your enzyme's not just flipped. Your enzyme's not controlled. It is deactivated, but it's not done. We can regenerate this. It can be regenerated out by 2,3-BPG. That's an important step. I promise I'll ask you a question about 2,3-BPG in step 8. Reaction 9. Oh, we're almost done. 9 minutes left. We gotta hurry. Dehydration of 2-PG. So we're left with 2-PG. Step 9 is a dehydration reaction. Dehydration of 2-PG to phosphoenol pyruvate. At this point, you should have, even if you have not read through all the steps, you should have seen phosphoenol pyruvate. To have a large, or have a big positive or a big negative delta G naught. Big negative delta G naught, right? Bigger than ATP. Bigger than ATP. This reaction is catalyzed by enolase. Catalyzes the formation of phosphoenol pyruvate. What's the difference in a dehydration reaction and a condensation reaction? What? Adding or losing of water? Yeah, they're both losing water. Right? What's the difference?
Is that when you take out the water to drive the reaction this way, like forward with the products, and the other one increase?
No, I don't think that's right. I don't think so. Specifically, I know the difference in a dehydration versus a condensation reaction. Is a remover of water. From one molecule as opposed to two molecules. In a dehydration reaction, your water is actually removed from a single molecule as opposed from an H of one molecule and an OH of another molecule coming together to form water. You have two H's and an O removed from a single molecule as opposed to two molecules. So, enolase reaction, what does this look like? Carbon doesn't do that. This enzyme is stabilized at the active site by two divalent magnesium cations. Oh, this is not 2-PG. This blank is drawn in blue because blue is water. These are the two, it's the hydrogen and the hydroxyl group that we're going to be losing through dehydration. This reaction is primed by an active site lysine. Acting as the general base, subtracting a proton. Red. Arrangement leaves us with a very stable intermediate. also has an active site. Glutamate, glutamic acid. Highly charged intermediate. You can see the need or stabilization of the active site. Our lysine is now protonated. It grabbed one of our hydrogens. Glutamate acts as a general acid to facilitate the removal of our water. Removal of our hydroxyl group as water. Our product, removal of water. Removal of water. Hydrogen. Glutamate, not our hydrogen from this carbon. Leave this with phosphoenol pyruvate. So a few points about this mechanism. Lysine acts as a general base to abstract a proton to initiate this reaction. Hopefully you can see the role of magnesium. Cations stabilize. Enolate intermediate. And the last step, glutamate. Glutamic acid. Acts as a general acid to facilitate the elimination of our hydroxyl. And we are left with phosphoenol pyruvate. We're out of time. We got far. We did. We almost finished. One last step. Step 10. Step 10, I'm not going to try to cram it into 2 minutes. Step 10 takes a little while because step 10 is your third and your final regulation step of glycolysis. Third and your final regulation step of glycolysis. What's up?
We said PEP was again possible.
Phosphoenol pyruvate. Phosphoenol pyruvate.
What was the second regulatory step?
Second regulatory step. First regulatory step was number 1, right?
Yeah.
Second regulatory step was number 3. Step number 3, phosphofructokinase. And if you need to know something about regulation steps, you need to know something about phosphofructokinase. It is the major regulation step. It is a major regulation step.
I'm sorry, repeat
Major.
number 3 once again. I didn't catch.
Phosphofructokinase. Yep. Step 1, hexokinase and glucokinase, right? Regulation step. Step 2, not a regulation step. Step 3, phosphofructokinase is a major regulation step.
Done.
Major regulation.
Meaning that you're going to be doing stuff at the very beginning of the